good afternoon. Have you ever wondered how changing hormone levels drive complex behaviors? Who are these molecular unsung agents orchestrating our lives? Over the summer, I delved into studying the endocrine mechanism behind female songbird regulation. So, in a recently published study by Dr. Odom in Nature Communications, it was found that female song is much more common than previously thought. It was previously thought that it was mostly absent. For decades, research on birdsong regulation and birdsong in general was mostly focused on males. There was a really high bro bias, as one would call it, and so we figured out that in males, high testosterone boosts singing, but we only know that females sing most when testosterone is low, and this is a very interesting question for us, because their testosterone is consistently low, especially when their singing rises to their maximum during egg laying and when they're defending the nest during egg laying. So in this figure from Dr. Odom's study, we can see that there are different levels above absent for female song incidence. It reaches rare, occasional, regular, even the same as males. So in one example, that pink bar over there labeled occasional, it corresponds to this model right here, which is a cartoon that represents all the species in that incidence level where they have occasional female song. This bird right here is actually the northern house wren, which is the bird that we study. And in the northern house wren, we were able to collect plasma samples throughout the breeding season. And what we're doing to answer this question, what hormones regulate female birdsong, is to quantify hormones in these plasma samples throughout the breeding season. So how are we going to quantify hormones? The traditional method was with an immunoassay like the ELISA, and there are several limitations to ELISAs because we can only typically quantify one hormone at a time. It requires a large volume of precious samples. There's a risk of cross-reactivity because of the antibody in an anti-sera solution, and sera hormones having very similar structures, as well as metabolites and precursors not even being detected at all. And with bird plasma samples specifically, they're very precious and volume limited, and even 50 microliters is very difficult to come by. It demands field work and handling very small birds with a lot of care. So this is where my project comes in. We're leveraging high-performance liquid chromatography mass spectrometry. It differs from immunoassays because it detects a suite of hormones at a time rather than just one typically, and it broadens profiling by including precursors and metabolites, and it maintains very high specificity and sensitivity even at very low sample sizes. So we use a deuterated internal standard for each hormone, and that kind of helps label and track the target hormone and accounts for sample loss even after processing to maintain accurate quantification. And this is like the instrument that we use. And with this project, we're hoping to expand the scope of the Odom lab to include hormone quantification with mass spectrometry to draw more insight and information on the biochemical pathways and neuroendocrinology behind female birdsong regulation, and perhaps behavior and endocrinology broadly across many species, including our own. Thank you.
Questions for Amanda?
Did we compare the results from the HPLC with the traditional ELISA?
So we didn't end up doing an ELISA, but for HPLC MS, it's more like separating by polarity, like physical properties, and then separating, getting exact mass to charge ratios. Whereas in ELISA or an immunoassay, we're kind of just looking for this color change. It's more like a qualitative analysis. So we don't actually have that comparative analysis in this project, but previously in Dr. Odom's lab, she has done ELISAs with different types of analysis for regulation and other chemical pathways.
To have sufficient
volume to do an ELISA. They have to pool the sample. This will allow us to get direct detection from a single individual, and keeping in mind this is survival. So we only take a small portion of blood. We're not, like, this is not sacrificial, meaning that the animal survives after the blood draw, versus an ELISA would require a larger sample volume, which would require pooling. So this would be allowing us to get a statistically significant circulating hormone level.
Thank you, man.